Good afternoon and welcome back everyone. With the spirit of show must go on, may I now request Dr. Meena Kanyalkar, Associate Professor of Pharmaceutical Chemistry, to introduce our speaker, Dr. Surajit Debnath, who will deliver the session on hands-on simulation on enhanced stability, engineered proteins with derived traits as therapeutic agents. Over to you, ma'am.
Thank you, Ruchira. Am I audible?
Yes, ma'am.
Thank you. So, good afternoon, everybody. It's a post-lunch session again. But I'm very much sure that our today's expert speaker over here is going to keep us attentive as well as dynamic throughout. He is expert from the area of protein dynamics and simulation, protein engineering, and he knows how to tune the protein and then simulate it for the therapeutic effect of it. So I'm very much sure that he is going to tune all of us by sharing his rich experience in protein engineering and simulation, followed by the hands-on experience with protein simulation. So I'm sure everyone have downloaded software mentioned by our speaker, and the link has been provided by the organizers yesterday to you. So I'm sure you are ready for tuning with him. So I'll take my pleasure to introduce today's young and dynamic speaker, Dr. Surajit Debnath. He is going to speak on engineered protein with desired traits as therapeutic agents with hands-on simulation on enhanced stability, the thermodynamic stability. Dr. Debnath is head of the Department of Medical Laboratory Technology, a government biomedical institute from Tripura. He has 13 years of experience in biomedical research and protein engineering. He is recipient of Young Scientist Award from various government funding agencies such as DBT, DST, CSIR, and also recipient of prestigious Tata Trust grant for CME course in Royal College of Pathologists, London. He recently filed 2 patents, the Indian patents, and he is guiding a group of international interns from Russia, Turkey, and West African countries. Dr. Debnath participated in various academic projects nationally as well as internationally. To name the few, the cytogenetic project at AIMS, biotechnology project at IIT Kharagpur, biomedical engineering project at IIT Guwahati, plant genomics at NB PGR. Also, he has participated in various research academic projects at institutes of international renowned places such as Germany, Denmark, and Hong Kong, which are sponsored by DBT, DST, and CSIR. He is also consultant for biotech companies in Western as well as North India. He has conducted more than 50 such workshops and chaired various types of national as well as international conferences. He is also reviewer for several Scopus Index journals published by renowned publishers such as Elsevier, Springer, etc. And it will be a great pleasure for all of us to know such a diverse type of research interest, research area where he has worked, and definitely we will enjoy the hands-on experience which will be shared by him. So the digital platform is yours now, sir. So over to you, Dr. Debnath.
Okay. Am I audible, madam?
Yes.
And my screen is visible as well, right?
Yes, sir.
Okay. So good afternoon. And as madam has said, post-launch sessions are very challenging for the participants as well as the speaker. So I'll go ahead. So the today's talk is on engineered proteins with desired traits. So when we are talking about therapeutic proteins, going to the next slide.
There is some lag
Great.
period.
Okay, great. So I'll go to the next slide. So what happens is when we are talking about therapeutic proteins, a protein for biomedical applications, we are essentially talking about three things. We are either talking about the diagnostic applications, then therapeutic applications, or as a vehicle for drug delivery kind of application. When we are specifying our applications of proteins for therapeutic purpose. So, and many times recently there has been some renewed interest in to combine the diagnostics and therapeutics both in a single protein. So opening a new vista that we today call as theranostics, where therapeutics and diagnostics are taking place hand in hand in a single entity. So in diagnostics, when you are looking at these are essentially for bioimaging and biosensor kind of applications. Now in bioimaging, these are mainly related to designed antibodies which are having what you call is some kind of chromophore or fluorescent tags which are usually useful for localizing specific kind of surface antigens present in different types of cancers to identify the stages of cancer, this and that. So when we are talking about biosensors, these are mainly some kind of redox group of enzymes which are involved in group transfer as well as electron transfer, which eventually give rise to a local change in the potential difference in a particular electrode, and they are helping to identify some kind of dynamic change that is going on inside the body. So biosensors and biosignaling are these two kind of proteins that are there. Now, when we are talking about therapy with the proteins, two major applications are regenerative medicine. For example, using fibroblast growth factor after a operation or intervention. For example, critical interventions like angioplasty and angiography, when you need to repair some kind of very minute blood vessels. Other than this thing, then at that point of time, this fibroblast growth factors gel, which is available in the market, are very useful. Other than that, these are cancer treatment, as all of us know. Other than the chemotherapy part, the peptide-based therapeutics that are there are for cancer treatment, and then the antibacterial and antiviral agents. These are also there as a therapeutic peptides. Now, drug delivery mechanism, where gene delivery mechanism is done through some kind of peptide vehicles, and then there are some applications where these what do you call as various other medications are also delivered through. For example, a very good example could be adjuvant. Now, adjuvant is also a drug delivery mechanism only, where we are delivering a particular subunit vaccine, which is having a very low molecular weight, and therefore it is getting masked and escaping the immune response and not able to generate sufficient immune response. At that point of time, what we do is we add an adjuvant, and therefore the adjuvant acts like a drug delivery mechanism in order to enhance the overall immune response towards a particular subunit. Because, for example, in any viral vaccine, many subunit vaccines are such that the overall subunit is so small that it is not able to elicit sufficient immune response. At that point of time, these adjuvants are also very useful, and therefore we can talk these things as vehicles. Now, these engineered antibodies also are in use today. Several examples, some common examples would be like for rituximab, which is an engineered monoclonal antibody useful for autoimmune diseases, as well as these days, recently they have also been repurposed. The drug repurposing is a very common thing these days. So this has been repurposed for glomerulonephritis, as well as what we call is this nephrotic syndrome in children. A very interesting study has come out. So rituximab was initially developed for as an what they call as to treat autoimmune conditions like SLE, systemic lupus erythematosus, and very serious cases of rheumatic arthritis. But today, I think it will be there in NCBI also, that it has been repurposed for mainly in case of children who are not responding to steroid therapy to get rid of their nephrotic syndrome, and they are losing protein every day and accumulating a lot of fluid in the body. So in these cases also, rituximab is an engineered antibody that has been used. Other than that, in breast cancer, that very famous application of Herceptin, a monoclonal antibody targeted at ERB1 antigen that develops due to breast cancer. So that has also been in use. Other than that, some immunotoxins, then immunocytokines, and antibody drug conjugates, these are already there. So one more group of therapeutic peptides that are in use, and so much of interest that has been developed recently are on interleukins. Interleukins, then interferons, and this group of peptides. So here I'd like to mention about PGI Chandigarh that has done work towards using interferon gamma for hepatitis B and hepatitis C patients. In India, they were the pioneers in this, and today they are an international protocol to treat what do you call is hepatitis viruses, the DNA virus infection of the liver, using interferon gamma mostly. Then there is a very common protein, protein, which is very much in use for dialysis patients who are having already got rid of their both the kidneys, and therefore there is a compromise of EPO, and erythropoietin, genetically engineered erythropoietin, is being used for their overall so that the CBC or complete blood count remains stable in these patients. So what happens is the metabolic wastes are getting rid every day through dialysis, but they have a very unhealthy what do you call as unhealthy blood picture. When you draw blood from this kind of patients, you see that the RBCs are immature and mostly with nucleus, and the main reason behind this is the lack of EPO, which is produced by the kidney. So in these patients, EPO has been given as an emergency medicine, and EPO, erythropoietin, is a very good example of engineered protein, and a very important one. These are the list of engineered antibodies and growth factor and signaling molecules that are either in full scale use by the clinicians and doctors, or it is in the pipeline to get delivered after the stages of clinical trial. So these are very fascinating stories where so much of work has been done, and in therapeutic peptide domain. So I shall change it to the next slide. Here again, some example therapeutic antibodies. Now these antibodies are mainly in pipeline only and not yet under this thing full scale by the peers. In this group, these are ADCC enhanced antibodies, antigen dependent cell mediated. Some recent developments. That now, what are these problems? The problems is that many a times it may happen that you will not able to get the anticipated result out. One such example would be times it has been there are some crosstalk between cellular machineries. So we'll do a little hands-on in order to understand that how madam, while the PPT is getting shared, shall we go to hands-on part so that you can share with the participant? First link, there is a link, madam, if you see, not the video, one NCBI thing, the fibroblast growth factor. Now fibroblast growth factor, as you will also know, that it is helping to regenerate the skin, and at the same time it is also a progenitor which stimulates skin cancer. So what happens is, if a particular protein you are using as a therapeutic agent, if in the long run it has some other counter manifestation, then what is, so we need to do something so that only the desired effect is there. And Particularly, one such problem is the stability of peptides. So most of the peptides, even the vaccines, if you see, the vaccines are costly because it is to be carried in a such a that it should be preserved in a certain way when you are transporting the vaccines to a faraway place. So, and therefore, what happens is the overall vaccine is not very costly, but the procedure and the other things becomes very. So, increasing the shelf life of a therapeutic peptide is also a very big concern. And we in our lab are trying to explore these areas as well. So here, Madam has shared one video which shows that how exactly a very famous and very common therapeutic peptide, that is insulin, is being produced in the body. So it will be helpful to see, not for the biomedical group of students, but teachers, but for the teachers from other background. So, Madam, if you play the video without any sound, it will be great. In mute condition, madam. Yes. Now this is the human body where in pancreas we are having a specific kind of cell where insulin is being produced. Now how this is being produced? Now there is a switching on mechanism which will stimulate the production of mRNA for insulin, and subsequently it will undergo a particular kind of mechanism translation which will ultimately produce the pre-pro insulin, a large peptide. So once the pre-pro insulin is there, it will undergo a autocatalytic cleavage to become a fully functional insulin. Now this is the interior of the cell. The insulin is being produced. If you see, this is the mRNA. Initially, it was the gene. On that gene, a particular kind mechanism will take place which will make it into an RNA, mRNA, then eventually the protein will be formed. Now this protein is nothing but the insulin. So what exactly happens is those patients who are not able to produce insulin of their own, they will be taking an engineered insulin. Now this is the gene of the DNA that everybody knows. And then there are some molecular cleavage mechanism, and it will cut down the whole genome into small pieces. Now this is the specific gene for insulin, where transcription will initiate, and it will become an RNA, and eventually it will be made into a vector DNA, which will be sealed by a ligase kind of enzyme. And the whole this machine, this is an artificial chromosome which will be kept inside a bacteria. Now this is a bacteria. The bacteria is developing, and eventually it is also producing the insulin in the culture medium. Now that insulin, yeah. Now gene has been incorporated into many bacteria, and eventually protein will be formed, which will be injected. Now see, it is injected into the body, and the patient who was suffering from hyperglycemia will be able to produce not its own insulin, but but an exogenous insulin. This is how a therapeutic protein comes in play. Thank you, madam, for sharing the screen. Now, madam, may I request you to please share the third link, unshare or close this screen, and go to the email, madam. Yes, no problem. You can share the email here itself. Otherwise, link, there is a link number 3, madam. Not an YouTube link. This is an NCBI link, madam. Thank you for sharing the exact thing. So, yes, madam, you can also share this link in your box so that the participants can also click on that. That you have exactly made the correct one. So, dear teachers who are already the participants and already are the teachers. So what happens, sir and madam, that one very important enzyme that is used for therapeutics in an indirect way to make most of the biosensors that are there, this thing system. All sensors use an enzyme which is known as cytochrome oxidase. Now it is a redox enzyme and it is isolated from a bacteria called Pseudomonas. Pseudomonas is a soil microbe, and this soil microbe produces, and it is also a aerobic bacteria. Aerobic bacteria means it can sustain the presence of oxygen, and it is also having an electron transport chain in its system. And this cytochrome oxidase is a part of the electron transport system. So there are not many which are having this particular enzyme. So what we are doing is, in the sense the fraternity is doing is, whenever a bio sensor is being produced, for example, to identify blood glucose in a strip, the strip that you use for home checkup of blood glucose, it uses an enzyme cytochrome oxidase from Pseudomonas putida. Now, how we can engineer a Pseudomonas which is thermostable? You need not to store it at a certain degree of temperature. If your strip, glucose strip, has to be stored under 44 degrees of centigrade temperature, eventually the cost of each test will increase to a significant extent. So what is the way we can reduce the cost? So one such this thing is to generate mutant of this particular cytochrome oxidase, which will eventually lead to an enhanced thermostability. So here what doing is the fraternity is doing is, they have made a whole genome sequence of Pseudomonas. Now this is, you are looking at the whole genome sequence of a particular variety of Pseudomonas. So, and this particular format is being present in NCBI, which is a global database of science information, including protein and proteomics and genomics sequences. Now this is the whole genome sequence, which is large thing. We need not to, we cannot directly use the whole genome, but a certain part. So there are methods by which we can isolate a particular site of the whole gene, which is coding for cytochrome oxidase. So we have done this already, and there are the protein sequences. So this is the first thing which will give you the whole genome sequence of Pseudomonas. And then, madam, I'll request you to share the next link to this, which is for the protein sequence of oxidase from the same bacteria, Pseudomonas. Yes, madam. So this is the whole genome which has been done in a shotgun sequence. Today, better sequencing technologies are there, which are, for example, Illumina, next generation sequencing, Roche platform, and several other, which will give you excellent result with high accuracy, and the whole thing can be sequenced in a sequence of the. So previously we had on the whole genome. Now we have entered into a protein data by clicking on a specific link. So those who are already accustomed to the database, they already know that there are inbuilt tools which we can do the transcription as well as the translation one by one. Other than that, we can also even search in the database to find out the specific gene sequence. Now this gene sequence is also very interesting because it is a complete sequence. And this format that madam is sharing now is called GenPept format. Now in this GenPept format, there are several things that could be known. That this is only the format where all the data is available, okay, including the sequence. So as you can see here, the locus name, this locus is nothing but the specific site of the genome where this particular gene, particular protein is present. And then so what will happen is, as you can see here, the length of the amino acid is also there. This is 57 amino acid, and there are the unique accession number, and then these are the author names. As you can see, authors. Then the title of study they made, and then several other references, journal name, and this and that. So many information is there, including the sequence. Now what happens is when you are using a simulation tool, you will be more interested in a. What we will do is we will change the format. So may I ask madam to click on FASTA, FASTA. If you can see after the headline of the protein name, cytochrome oxidase, there is bank accession ID, then there is identical proteins, and then there is FASTA. So madam, may I request you to please click on FAA. A blue color, this can be seen. FASTA. If you can click on, yes, madam, absolutely. So participants can also click on FASTA, so that what will happen is, yes. Now this is the FASTA sequence. Thank you, madam. So as you can see here, much of noise has been reduced. So what we did here is kind of format changing, which is inbuilt to the tool that we are using it. So here what has happened is the complete sequence of this particular protein is there. Now this is the primary sequence of this protein, which started from M. M stands for methionine. And so you are very much sure that your N-terminal end of the protein is secured. Usually what happens, there are some cases where you will find protein sequences, but many a time it is a truncated protein. So if it is a truncated protein, it will be a problem for you, because what will happen is you will not get the active site of that. So here it is a complete protein. We can think at least the N-terminal end is okay, and the rest you need to have some background information that how many amino acid long is the Pseudomonas cytochrome oxidase. Now this is the wild type Pseudomonas cytochrome oxidase protein. Okay. And as you can see here, the FASTA format starts with a greater than, then there is a accession number, then there is the name of the protein, and then the name of the species as well as the name of the strain that is there. So this is the whole protein sequence of the enzyme. Now this protein has been coded by a DNA sequence which was there in the whole genome sequence. So what you can do is, if you have a specific knowledge about the sequence, you can directly copy the sequence from the whole genome and paste it. Otherwise, you can search in the search window of and you can end up with this particular protein. Okay. So we are having the wild type protein now. Now this wild type protein is such that you need to preserve it at certain temperature, because the optimum temperature for what do you call is Pseudomonas putida growth is approximately 25 to 27 degree centigrade. So many a times what happens is you store your glucose strips that your AccuCheck or things like these medical devices at a temperature which is lower or higher than this. And one more problem with this particular wild type protein is that it is having a certain half. What I'm trying to say is its expiry date is not very large. So if I use what do you call wild type cytochrome oxidase is in my electrode. So what will happen is it will only be 1 month expiry date. So it is not possible and not viable for an industry. So what the industry will try to do is, the industry will try to maximize the time, isn't it? By increasing its shelf life. So how exactly it will do it? So there are several other techniques that to increase the shelf life of a protein. So most of you have already used this methodology. One very common methodology is to entrap a protein into a matrix. The very most common practice to enhance the stability of a protein is to make it into a which is very common practice among pharmacy as well as biomedical other departments, to make it into a hydrogel. The gel is helping the protein to withstand the unfavorable environment, and when you apply it, it gives you a sustained release kind of effect, because the matrix is there which will be solubilized after, because many a times the matrix will be from some inert gel, like it could be some kind of polymer, or it could even be some kind of a petroleum jelly which will respond to certain degree of temperature, and that it will melt and the protein will be delivered. So it will give you a sustained release effect as well as it will preserve the active site of the protein. So this is very, very common in terms of, shall I say, the growth peptide, as I have said, for minute, for example, some laparoscopic surgery if you do. So after the laparoscopic surgery, they'll make a bandage. So before the bandage, they'll add the growth factor gel. And one more very, very common that I forgot to mention, which everybody will recollect, that there is a medicine in the market. If you go any shop, you will get a Plantex gel, okay, or Placentex. There are two trade names. Now, Placentex is very, very common. Any shop will have Placentex, an excellent example of entrapping peptides for enhancing its stability. So, out of many such this is very, very common in practice. Now, Placentex is used in order to treat, okay. Now, bed sore is a condition when a patient is constantly lying on bed and several pressure points are being developed in the skin, and therefore the skin becomes fragile and the patient generates some kind of inflammation in the skin. So the best medicine till now today is Placentex. Anybody after your session, if you go to your next medicine shop and ask for Placentex, you will get a approximately 200 to 300 rupees gel. It is nothing but the placenta extract. Okay, excellent example. I forgot initially, and very, very common example as well. How growth factor peptides are entrapped. Now in placenta, what happens is, as everybody knows, placenta is where it will ultimately grow into a full-grown animal. A lot of growth factors are there, all kinds of growth factor in fact, including fibroblast, then epidermal and whatever you name for. So what happens is they'll isolate the placenta of usually an animal, a mammal only, other than human, and they'll homogenate the whole tissue and into a hydrogel. Okay. So, and it is sold as Placentex, and a very good medicine for the geriatric patients suffering from bed sore. So what happens is in the bed sore, if you apply it and make a bandage, so what will happen? You will see that the bed sore is getting cured in a very short period of time. So what does Placentex contains? It contains the homogenate of placenta. That is nothing but the fibroblast and epidermal growth factors, which are entrapped in a gel in order to preserve its shelf life. So similar is the case here. If you want to preserve the shelf life of this oxidoreductase, you need to do some kind of adjustment. Now here the problem with biosensor is that you cannot use this kind of this gel kind of thing. It is useful for topical applications or local applications, but not for a electrode. So what these electrode people will do, they will do a process called so what they'll do is they'll take the DNA from the holome, as we have seen in the video, that from the holome a particular site of the gene will be identified and it will be cut down by a restriction enzyme, and then what they'll do is they'll put into a PCR, okay, in order to clone the whole particular gene. Now this PCR will be done in a method which is called error prone. Error prone PCR is a PCR where you have some wrong thing to happen with your PCR product. So what will happen is, many a times it will happen that some kind of mutation is incorporated. So next your job is to identify all the clones and make it into an expression vector and put it into a host cell, as the video has explained. Eventually, it will produce different types of proteins, and then you need to screen these proteins, which is your suitable protein. So this is one thing. One more approach, which is also PCR, very, very easy. I have today morning, I had an opportunity to speak with the coordinator madam, and she is having a very interest, so much of interest in protein biology. So many of us can relate to this. It's a very simple PCR where you are pre-designing a primer. In that primer, you are already incorporating a mutation. Okay, and then what you are doing is you are simply cycling the product, and you are ending up with a gene which is a little bit mutated. 1 or 2 nucleotides has been shifted. What is the effect of this mutation? It may happen that inside, but all the other proteins are present in such a way that end up with a more thorough. Be able to know that in Scandinavian countries they are using this what do you call it, saturation mutagenesis randomly, and they're even getting excellent results. I give you one more example of a protease which is useful for making detergent powders. So all of you are well accustomed to the detergent powders. Now in this detergent powders these days we are having peptides, that is like proteases. Now these proteases are so strong that they like increase their activity 52 times, 52 times just after a single. So this is the extent. The Scandinavia mainly, I had an opportunity to interact in Scandinavia and Western Europe. Danish companies, Novo Nordics, these are very, very good companies. Much in the forefront in their Novo Nordics and then one more. They are only. So these companies are doing serious and getting excellent results. So conceptually a very tough job, and you can and today these mutagenic kits are also available from Indian, so many Indian companies. So it is not a big deal. But the deal here is where how you determine where you need to incorporate the mutation. So in order to finalize the exact specific site where you need to incorporate the mutation is the like the $1 million question. So in order to answer this, smaller labs like us, in our case, we are very much having so much of shortage of funding, and therefore we cannot afford using a kit with some wrong results. So before doing an experiment, we have to think not twice, 10 times that where exactly we are going to incorporate this mutation. So today, computational biology has helped us a lot in order to understand that where exactly you need to put the mutation. So I'll request coordinator madam to share the link of the next this link, which starts with HTTPS, then Harvard dot polyfen2 one link. It will be shared to the chat box with participants as well. If they wish, they can also click and enter into a grid computing system of Harvard University. That is nothing but a supercomputer facility which we will use locally. Downloading any software, nothing. So there in computational software space also there are. Yes, madam, exactly. And I'm very much thankful that you are coordinating in a nice way. So this is Harvard University we entered into, and after putting specific what do you call is commands, we will enter into the Harvard University supercomputing system. So before that, what we will do is, as you can see here, it's a database, not exactly a database, a tool which uses machine learning capabilities. So machine learning is a concept where you are training your to predict some outcome. Okay. So according to the computational principle and computer scientists, there are various kinds of algorithms that the computer can understand and also learn if you a training set. Okay, so there are two aspects of, there is a software or an algorithm, and there is also a database. Now the software is designed in such a way that it is adapting software. Okay, now these adapting softwares are generally called machine learning algorithms. So one such machine learning algorithm is called a probabilistic classifier algorithm. Okay, so I will not go to the much detail of this, but you can definitely Google these terminologies if you are interested. The probabilistic classifier is nothing but a classifier which will have its own experience from previous events, and on the basis of the previous events, it will try to predict an outcome. Okay, so if you go to this tool, and on top, if you see, you will see WESS.db. I do not know how many of you can see it. just after the polyfen 2, you will have home, then you are having about, then you are having help, you are having downloads, then you are having batch query. Yes, madam, do not click it. Do not click it, otherwise we will enter into a very big database that I don't want. So this is the database, sir, madam, where the training set is there. Okay, so if you wish later on in your free time, you can go to the database and enter into the it's nothing but a database of mutations. So the concept is so simple that anybody can understand. So what it is doing, it is having a prior experience of all kinds of mutations that are available today for everything, for human, for animal, for bacteria, everything. As much they can accumulate in Harvard University, they accumulated the data in this WHESSDB. The extension is .db, means a database. It is nothing but an Access file, Microsoft Access kind of file. So what it is housing? It is housing a database of all possible mutations and its effect. Okay, but they have designed a software. The software is such that on the basis of the experience in the database, it will predict a result. Okay. Now, what is our problem? Our problem is this, what we call cytochrome oxidase, a more stable one, so that it can withstand a shelf life within degrees for not less than 6 months. क्योंकि ये basically 1 month के लिए होगा, तो ये उसका फायदा नहीं मिलेगा कंपनी को. क्या चाहेगा? It will try to enhance the shelf life as much as possibly do. So we want to do a mutation on the cytochrome oxidase protein by using our saturation mutagenesis technique and PCR and like that. But the problem here is we do not know where exactly we need to introduce them. So this is where machine learning will come for the labs which are suffering from severe ink crunch like mine. So I cannot able to do 10 different tests in a single day to identify a particular amino acid, because they'll end up not less than 25,000 rupees for 10 different tests. So I cannot able to afford this. So what I do is I use a machine learning approach to identify what are the specific sites. So what I do is I reduce the number of false positives. Getting my point? So my objective is to use computational biology in order to reduce the false positives that I can end up. If 10 experiment करेंगे, उसी के बाद पता चलेगा कि चलो ये काम करेगा. So if it is like that, 5 different experiments में मेरे को पता चल जाता है, so it is very good. So this is our objective, to use machine learning in this case. So what we will do is we will identify suitable site which will help us to incorporate a mutation, but not destroying the protein itself. It can happen that you are introducing a mutation and all your activity is lost. This can also happen because the protein is having a particular active site, and if in the vital amino acid you are substituting, for this particular cytochrome oxidase has a heme prosthetic group. ये heme जो prosthetic group है, ये positively charged है, तो उसको उसके लिए तो negatively charged amino acid चाहिए होगा उसके चारों ओर. तो आपने क्या किया, glutamic acid residue को substitute. Glutamic acid was very much instrumental to make the coordination bond between the heme and the protein, because cytochrome oxidase is a hemoprotein. If the heme is lost, your protein is lost. So you cannot afford to. So you need to have sufficient background information of the protein structure. At the same time, you also need to use these machine learning tools in order to find out where exactly you will do the mutation. Okay. So what I'll request, madam, is, madam, may we go to the last protein sequence that you shared with our participants? The protein sequence, madam. In the same window, you do not close it, madam, because we will come to this once. So we will go to the protein sequence. Not this, madam. It is already there. Yes. So we are already having the protein sequence. Okay. So, madam, I request you to copy the whole protein sequence from the greater than sign to the last digit of E. From the greater than sign. Yes, madam. Please copy. The participants, Saran madam, if you wish, you can also do this. Yes, madam, fine. Now you please copy it, left click, and then go to polyfen. Yes. Okay. Now Madam, please paste it in the FASTA format. Protein sequence in FASTA. Second window, madam, not fast. Second window. Yes, madam. Yes, madam. Yes. Thank you, madam. Thank you. What we are doing, we are taking the sequence into the window. Now we have given the instruction to the algorithm, which is probabilistic classifier. The common name of probabilistic classifier algorithm is naive Bayes classification or naive Bayes algorithm. If you can Google it, you will get plenty of information out of it. I am not discussing it. So, and the main importance of why we are using, there are plenty. For example, artificial neural network, then there are support vector machines, and there are a couple of other things. So why we are using a particular kind of algorithm? This is, so there are some things that to be understood. So naive Bayes classification, the output is such that it not give you a 0 and 1. That means it is not a binary result, yes or no. No, it will give you a probability as an output. The output will be a probability, and on the basis of your knowledge, the user will take the decision. Okay, हम देखेंगे अभी. So what will happen is it will be a helping. You will get the help, but the ultimate decision you will take on the basis of many evidences. Okay. So you take your decision, a confirmed and well-informed decision. They will not directly dictate that you do this, do that. No. Therefore, this is the beauty of this particular algorithm. If you click on some other and Google the particular thing, you will come to know a lot more. And many participants, sir, madam, who are from computer background, they know much more than this, whatever I have discussed. So, madam, what we will do is, we will substitute the 8th amino acid. So, madam and sir, if you can see, these are M, I, R, G, L, G, T, V, V, V. So, what is that? Methionine, isoleucine, aspartic acid, glycine, leucine, glycine, threonine, valine, valine. So, these are the amino acids that are present. Okay. So, what we will do is, we will replace the 8th amino acid. So how do we replace it? We are doing a dry run. Okay. So we will see if we mutate the 8th amino acid, what is the result? We are interested in that. So, madam, may I request you to in the position window, please write 8. Digit 8. Yes, madam. Position. Yes, madam. 8. Okay. Now what has happened is our 8th amino acid is a valine. So what we'll do is the substitution pane is there where A1 is there. So madam, from AA1, please select V. The last amino acid is V. WYV, madam. Please click yes. Yes. Okay. So what happens? We shall also think about this probability that valine is a highly non-polar amino acid. Okay. So highly non-polar means what? Means it is not having any polarity. Usually, a protein structure, it will be present in the interior where there is non-polar region. Means what? Means usually valine will be present in an interior core area of the protein, or if it is present on the surface also, it will be in such area where it is the protein is fusing with a membrane. Membrane is made up of also lipid. So lipid is also a non-polar region. So if this particular protein is a membrane-bound protein, which is also a membrane-bound protein, and it is embedded in mitochondrial inner matrix. So what happens is where the valine you expect valine to be present in the interior, or if it is present in the exterior part of the protein, it will be definitely in close contact with the membrane, because membrane itself is also non-polar. So you are replacing a non-polar entity. I'll try to replace it so that you get an excellent result of this particular tool. Not for the mutation purpose. The mutation purpose, you have to do several cycles of this permutation combination. Just showing you a single case, we will be using this particular substitution. So what we are doing is we are replacing. Madam, please come to AA2 and click on this arginine. Arginine means R. Madam, please click on the AA2 pane. The second amino acid is R. Yes, arginine. Please click on it. Okay. So now what we are doing, we have reduced the cost of a saturation mutagenesis wet lab experiment to a great extent by only clicking on this particular toolbox. So here what we have done, we have replaced the valine with an arginine. Now arginine is a highly polar receptor. Also, it is bigger, 2 times bigger than valine. Valine is a very small amino acid, not very big. In comparison to valine, arginine is a large molecular weight protein amino acid. At the same time, it is highly polar. It means it is having 3 different like positive charges. Okay. So what happens is now what happens is you are having a substrate where you are expecting anticipatory results. So you know that if you substitute a non-polar entity with a completely polar residue, there will be definitely some large scale what they call shift in the microenvironment of the protein. At the same time, some geometrical instability is also there. So given an opportunity, I will not lose the chance to discuss about geometric assembly of a protein. And I must mention Professor G.N. Ramachandran, a great Indian scientist, direct student of Sir C.V. Ram, who have proposed for Ramachandran plot during 1950s, just after the independence. And those who already know about it, it is great. But many of us do not know that even today, when we are designing peptide therapeutics in 1 year, जैसे कि COVID-19 का केस हुआ, उसके 1 year में we have ended up with many different vaccines. So it was possible because of computational biology and the structural biology fraternity over the year have not able to substitute Ramachandran plot till now. So till now, Ramachandran plot is an instrument tool which help proteomics people, structural biologists, and overall the biology domain to a very, very significant extent. And we should feel sorry that Professor G.N. Ramachandran could not get a Nobel Prize, which he truly deserved, even after his long gone. But he have died nearly 1979 or kind of. But when posthumously also, he deserves the Nobel Prize for sure. Even today, this is possible to generate a subunit vaccine within a year, and if all the companies, including Indian as well as foreign companies, come out with this vaccine idea, definitely in the pipeline, some or the other way, they have used Ramachandran plot for understanding the structural geometry. So you're telling here is other than the ionic microenvironment, the structure of the protein also plays a major role. So it can also happen that in the wild type protein, the 8th amino acid, until and unless it is a valine, it will be within Ramachandran allowed regions. So when you substitute the protein with arginine, you are making a rapid shift in the Ramachandran plot and geometrical analysis also. It may happen. So before doing the substitution, so many other things are also to be taken care of. So it is a very simple and straightforward idea because our the scope of this whole session cannot be a full-scale workshop other than a discussion session. So we are not discussing those things, but it is definitely there. While doing structural biology without Ramachandran plot, every study is incomplete. So what we are here doing is we are substituting valine with an arginine, and now we shall submit the query, madam. Now please submit query, madam, on my behalf. Yes, madam. I again thank madam for doing the great this thing coordination part. Madam, now we have entered into the grid gateway, as you can see here, grid gateway interface. Now grid computing is nothing but a mainframe or a supercomputer where it is doing a very large scale integration and very large scale calculation in a fraction of second. So here you can download the whole Polyfen 2 software in your local computer, but for that you need to have a NVIDIA workstation that Madam uses for molecular dynamics. So we want to reduce our cost, and therefore we have never downloaded this software. Other than that, we are using the grid computational pathway only. So this is the best part of it, that you need not to download a local copy. Instead of that, by using internet facility and from the remote server itself, you can do your calculations. So the position is 2. It is still pending, I think. Yes, madam, please click on refresh. Okay, see, the result has come out. So it has cross-checked and cross-validated the result, did the plot generation and in like maximum 15-20 seconds. So now, madam, please click on result. That means view. Yes, madam. Please click on view. So participants can also click on view to see the results. Okay. Now here you see. So what we did, we used the position 8, AA1 was V and AA2 was R. The description was cytochrome oxidase. Then we are doing the prediction. Okay. So here, what is the result? The mutation is predicted to be. They are not dictating you all over again. They are predicted to be possibly damaging with a score of. So here there are two things. The score is 0.922 with a sensitivity of 0.81 and specificity of 0.94. So what does it means? That it is potentially damaging. So this is an expected result only. Nobody will do a mutation like this. Okay. You will never do substitution of valine with arginine, because you know that it will end up with something where there is no value in that, and eventually your work will be ended. Though what will happen, the protein will become an inclusion body and it will be removed by the phagocytes, as well as your lysosome will come and destroy the protein because it is not conforming as per the Ramachandran plot. So Ramachandran plot is so good that the living body also follows these instructions. And it's a tremendous achievement, and we must be like amazed that how with a single protein he have achieved this high high scientific, and not one this discovery I think deserves 2 Nobel Prizes. Anyway, so what happens is this particular mutation is highly damaging, and as we have anticipated. Now, if we change these mutations by some other, for example, valine is there, and if we replace it by glycine, which is kind of similar, what we call as amino acid in terms of ionic environment as well as size. So, or leucine, which is exactly same in terms of its ionic behavior. Valine and leucine will be similarly placed. So in that point of time, you will see that it is not possibly damaging, or it could even be benign. That means no problem at all. So you have to look for each and every benign mutations and then put it into a structure, then make the structure verified by several tools, including Ramachandran plot. If Ramachandran plot says that it is within the allowed range, then only you can be sure that this protein can be expressed by the host cell. As you have seen in the video, madam has shown you that the expression vector should be kept into a E. coli, isn't it? Now E. coli is nothing but a host cell. Now if the protein is not within the Ramachandran plot, the host will not able to express it also. It will be expressed, but it will become an inclusion body, and it will get eliminated by internal proteases and all, because it is not folded in a proper. So this is the beauty of this structural biology. So this was the first hands-on that I tried to this thing in the session. So it gives you a very brief very general idea of protein engineering and how it could be helpful. So if you find out 10 different mutations which are benign, so what does it mean? It means that there are possibly not a very large scale changes in the protein, or out of this 10, at least one can give you a protein which will be a mutant, but it will preserve its vital characteristics. And then you have to do a molecular dynamics to see how the behavior, dynamic behavior of the protein. Okay, so I'll request coordinator madam to please play the second YouTube video in a mute condition. Second YouTube video. First YouTube video we have madam has shown us. The second YouTube video from the email, so that our participants can understand what is the dynamic behavior of protein. Yes, madam, in a mute condition. Okay. So this is a dynamic behavior of a protein which is very, very unstable, madam. So why it is unstable? Because we replaced this particular site or a domain in such a way that it becomes fragile. So how you simulate the fragility? By using several force field method. A very simple force field method which is useful for small scale simulations in a prolonged period of time is called normal mode analysis or elasticity analysis. Okay, so in this what happens is you are making some kind of dynamic movement in the protein in a particular simulation box in order to find out what are the fragile domains in the protein. Okay, so what does it mean? If you see carefully during the simulation, the whole protein is getting lost. I'll show you the intact protein also in our next simulation. So here what is happening? You have made a change, but the change was so bad that your protein is getting lost when it is coming in the environment. Now in environment, what are there? In environment, there are so much of stress. Ionic stress, van der Waals stress, hydrophobic and hydrophilic stress, so much of stress. The protein is having so much of problem here, isn't it? Now madam, please go to the second video. That means the third YouTube video, madam, from the link. The next video that you have shown us. This particular video, the same protein with a small adjustment. Now here you see the domain motions are there, but it is more intact than the previous one, isn't it? The alpha helix is been seen. Can you see? So what we have done here, we did a substitution, but the substitution is such that it is having some more, what you call, it can withstand some more, what you call, dynamic environment, isn't it? So this is how you need to incorporate so many different things in order to ultimately end up with a therapeutic protein which is having a desired trait. So this is how we can do several calculations by simulating the whole process, and ultimately after using so many things, ultimately you can come up with an idea that how good is protein is. What is the objective? The objective is to make it more suitable for therapeutic purpose as the people are doing. So one good example could be like if we can generate a vaccine, peptide vaccine, which is useful for all the different mutants that are there today. So initially we had Covishield and Covaxin, which was doing wonders according to our reports, clinical trials. But when we with several other what you call as mutants, we saw that instead of taking 2 jabs, people are getting rid of their lives. Why? Because the vaccine they took is no longer useful. This we call as okay. It is no longer useful for these mutants. So is there a possibility that we can generate a subunit vaccine which is useful for each and everything, each and every mutant, which is not possible, which is next to be possible? So in this case, what is your option? Your option is a customized vaccine. Whenever a mutant will come, you have to take one more jab. Whenever a mutant will come, you have to take one more jab. Now this whole scenario gives us an idea that we are looking into in future we need to think about precision medicine. Precision medicine is that the single medicine will not be helpful for everybody. So it should be customized, like you customize your dose. For example, a full-grown man, you are giving 600 mg of paracetamol. But for a kid, you are giving a suspension which is hardly containing 50 mg of this what you call paracetamol. Also, you are putting some more adjuvants so that the hepatotoxicity is reduced. Because many times paracetamol could also be potentially hepatotoxic. In order to reduce this, you will add some more like some amino acid into the system so that it becomes more tolerable by the kid. So you are doing a customization for that. Today the need is to customize each and expect this kind of customization within next 50 years, because the situation will be such that a single protein cannot help you and you need to do the protein engineering more rapidly. So by saying so, I'll go next to the next hands-on permits, and may I request Madam to please share her desktop. Click on the icon that yesterday we have all tried to incorporate. So, Madam, if you all of the participants also, if they can click on their desktop, this particular window will come. Now, this kind of window where we are doing everything by clicking and not writing the code, we shall call it a GUI, Graphical User Interface. Now, in this GUI, there will be several menu options. On top of the menu, there will be the file menu. So here the beauty of this particular tool, this is written in C++ and art is written in Python. So the beauty is that this particular software is very small, but its capability, if we can use it properly, is it can be used in some very good purpose also. Those who know about molecular graphics and design softwares, they know that a simple desktop is not possible to run high-end simulations. But this visualization tool is very small and anybody can do it. Madam, may I request you to one more time click on the same icon. So two different windows and simultaneously two different simulations we will do, and so that we can what do you call compare between the windows. Yes, one more time you click and minimize it. तो मैडम दो विंडो आएगा, सेम GUI, two window will come. तो थोड़ा मिनिमाइज कीजिए मैडम. हाँ, दो विंडो आ गया. वेरी गुड. So in the first window, madam, please load the first PDB that we have given you. So go to the file option, madam. Yes. Yes, madam. Then go to open. Yes. Please go to open. Yes. Now here, take it to your desktop. I think you have kept the data. Fine. Great. Madam, please click on the first one. Please select the first one. L1 ASP. Okay. Great. Fine. So, madam, now go to the second window. No, not go to the command. Go to the second window. One more window is there. Yes. Madam, here you please again. I am thankful that you are coordinating the whole process in such a good way. Second one, madam. Yes. Okay. If the participants want, they can also. So in two different windows, two different versions of the same protein has been loaded. Okay. So, madam, now may I request you to go to the display window, madam. Yes. Here, madam, if you go and you will see the molecular surface, bottom option. There are several options. I am not demonstrating those. I'm just going to the particular thing that I want. Molecular surface, right, madam? Yes. You click. And it will take a little time because the calculation is slow, because it's a great. So now, madam, go to the second window in which the second protein is there. if the participants want, they can also do the same. Yes, madam. Here also, please go to the display button and click on molecular surfaces. Okay. So if you can, we are not able to see it. Madam, if you can smaller these windows, both the windows will be simultaneously. एक ही साथ पूरा स्क्रीन में मैडम दोनों विंडो दिखेगा. देखिए अगर ऐसा होता है तो बहुत अच्छा. So they can instantly identify our particular. बहुत बढ़िया. Great. Thank you, madam. This is what the anticipated. मैं भी होता तो इतना अच्छा शायद जल्दी नहीं कर पाता. Thanks a lot, madam. So now, madam, the participants also can see both the window are showing you same protein only. It's the same, only a small change was there. So now we have to identify what are the change and how it has affected the protein. So now, madam, I'll request you to go to the color window. Color. In any of it, yes, go to color and go to temperature. You come down, do not click on anything, just click on, yes, please click on it, madam. Okay, it will do a little calculation and it will come back. Okay, great. Madam, now go to the second window. Yes. Here also color and temperature. Yes, the fifth one. Great. Okay, it will do a calculation and it will come back. Okay. So now here you see. Now here what we did, we substituted two different amino acids. So the first time was, if you can remember, in which we substituted a particular amino acid. In the right side, we substituted a different amino acid. Now, what Rasmol does is, once it generates a whole 3D structure of the protein, there are better visualization tool, but these are high-end and you need to have some custom software, hardware even. But this software is so small and so good that by a click only you can do many simulations. So here what we are having, we are having the same protein in two versions, and now we are looking at an energy landscape. Okay. Now energy landscape is what? Energy landscape is nothing but the delta G calculations. Okay. Now delta G calculations are nothing but what do you call as the amount of free energy that is there in the protein. Okay, any biological entity or any entity is having some kind of free energy into it. If the free energy magnitude is in negative terms, it means that it is stable. Okay. Now what stability means? Stability means that a spontaneous process will be there. The stability will be spontaneous. You need not to do anything. If your delta G content is positive, that means in positive magnitude, so you can think that your stability is reduced. Now, why is this? This is because, according to the thermodynamic principle, delta G is nothing but inverse relationship with delta S or entropy. Now, entropy is nothing but degree of disorder. So we call it delta G equal to delta H minus T delta. Very simple terminology which clearly indicates that if delta G content is less than 0, the degree of disorder is also reducing. So if degree of disorder is reducing, you are ending up with a more stable end. Okay. So in order to simulate this kind of energy landscape for protein, the software that is available in the market is highly, the free tools are also high-end hardware, but this particular software, which is maximum 25 MB of codes and very user friendly, can give you the idea of this energy landscaping in a 2-hour session for 5 minutes only. We are talking about this, and I am sure that some participants definitely got some. This is the beauty of this small code. So what it is happening is the right side, the blue color indicates that the protein is highly stable. But there is a problem. As you can see, there is a horn which is red. It means that there are some problem in this region. But get rid of this particular thing, your protein is highly stable. But in the right side, you can see several different colors, but you did the same coding only. What you did, you told the whole protein to give you a color on the basis of the temperature. Temperature is nothing but B factor. B factor is nothing but an indirect entity that quantifies for delta G. Okay, not delta G, but it indicates or it stands for something. Okay. So what you are having two versions of the same protein, one highly stable with a small abnormality which you can get rid of by mutagenesis or by truncation also. If this is there, you can also design a particular peptidase or protease which will get rid of this particular hormone. But the left side is you cannot be sure of. If you make this subunit that particular mutant, you will be rest assured that you need prolonged, what do you call is, you need prolonged refrigeration to transport this particular vaccine to a significant distance. But the right side, the blue one, is such that there may be some structural problem, but if you can get rid of the structure, you will be rest assured that your vaccine will be more stable. Okay, so we are doing a simulation. You cannot be 100% sure. It depends many other things, as I have earlier discussed. So this is how protein engineering can come in here. So what does it mean? It means that the thermostability is a very, very important question in proteomics and protein therapeutics. And before introducing the mutation in the protein itself, if there are tools which with the because we desperately need economy in setups like ours, where we are this thing starved for funding. So in this case, what happens? Depend on these machine learning tools to a great extent, and eventually you can also what you call is end up with very good what you call as a prediction. So in the first tool, we understood that how sequence substitution can be thought of, and next tool we saw that how structure based what do you call it tools can help. So it is not exhaustive, but there are plenty of other things that needs to be discussed when we are discussing about therapeutic proteins. But the objective of this session was to give some brief idea about what exactly we can do within our limits. And more this thing is there are shortage of tools and techniques, even hardware, where I could not share the PPT where we have discussed in direct this thing that what exactly currently we are doing. We have isolated two small molecular weight peptide from fermented local stuff, and we have purified it to a to some extent, and we have able to predict its primary amino acid sequence, not by sequencing or this thing, but by some kind of homology, and by some wild prediction also was in. But we come across to the thing, we are not having a single tool which will give us all things all together. So for last 4 years, we collaborated with a small this thing to make our own tool, which comes with a particular small hardware, and we have submitted to Indian Patent Office, and some rounds of communication has been completed, and we are very much anticipating that we will able to monetize our innovation as and we are also talking to small scale industries which are having exposure edutech ventures, if we, and we are talking to these people, and some have come forward to give us some small funding, and a small this thing has also been done by IIT Delhi, which have given us some and in this particular domain. So we are also very much open to, today morning also we have spoken to our madam that we are very much open to collaborations, like-minded labs where more sophisticated facilities are. We will be doing equal contribution of intellectual property, effort and time, so that we can eventually end up with some significant value addition in the existing things that are going on everywhere. By saying so, I'd like to conclude, and I'll definitely like to take some questions, and I'll be happy if I can answer them if it is possible.
Thank you so much, sir. I know that there were a lot of technical glitches were there in between, but with the help of
I'm very sorry
our
for that.
organizer, Madam Dr. Sane, Madam,
Yes,
they
excellent.
helped you go ahead, and you had really
Madam,
yes, sir. Hello.
coordinator madam has done a job. I'm very much thankful, Mrinal madam. My words are small. Thing you have done for. Thank you. Thanks a lot.
Yeah. So I think the show must go on. So we have, all of us have that attitude, and because of that, we have went very smoothly, even though the things were not working in our favor. So thank you, Mrinal madam, as well as sir. To summarize today's what we have understood from whatever your presentation is, basically you have given a very nice thought-provoking idea about the protein engineering, like from diagnostics to what you can say the therapeutic to the delivery, drug delivery. This protein and peptides are very integral part, and it is really upcoming things. We have seen that its importance when the COVID crisis came, and the lot of such type of therapeutic agents were required, whether it is a vaccine development or whether it is a development of lot of antibodies, antigens, and all such type of things. I think protein engineering is where we should look forward. During this entire session, sir, you have given a very nice two important tools you have taught us. That is, first thing was obviously they are machine learning softwares. One we have seen, that is Polyphene 2, which is like it's again it's a freeware, we can call it as, because you are using a web usage of locally contacting to the supercomputer at Harvard, and that has helped us to understand how to understand the mutation. Suppose if you are looking for a molecule, say example, you have given the very nice example of a biosensor, and if you will just try to use the your own biochemical basic knowledge, you can actually identify whether you can generate something which is possible or not possible type of mutants, because these mutants are nowadays making or doing a lot of havocs, and definitely just looking at screening with the database from such machine learning databases, it will be definitely give a breakthrough for further research. Definitely it was a very indeed nice interesting experience, and I'm sure definitely I will go through that Polyphene to learn more. And I'm sure our audiences will be willing to work on that same tool. Second thing is the simple freeware Rasmol, which is very much available. It is very small comparatively software, which can give a brief idea about what type of mutations or what type of changes you can do in your peptide. So that definitely bring the interest in your work, or it may give a brief glimpse about how much changes you should do in your peptide. So all this simulation can give you a basic interpretation and prediction when you are moving ahead with actual protein engineering, because that is one of the costly, time consuming type of work. So definitely this glimpses will help us to understand how the mutation, like especially the people who are working in the area of pharmacology, biotechnology, they can get a glimpse because many times the simulation, computer aided drug design becomes a taboo, and they don't want to work, like they don't want to try this. But definitely these small things can help them to enhance their confidence in this area. At the same time, sir, we are giving our best wishes for your patent. Like, you should definitely get the patent for the new software or the new link you are creating in this era of protein engineering. So all the best to you for that. And it was really indeed interesting lecture. I have got a lot of notes in my hand now to ask you questions, but let us take some questions from audience if they are.
It was a very
Yes, right.
Yeah, Mrinal.
So I'm a pharmacologist. I got an opportunity to learn about chemistry. So thank you, sir, for giving me that opportunity. And as a coordinator, I
Yes,
would like
thank
to
you.
apologize to the participants for any delay in the session happened because of some technical. But I think we have managed.
Yes.
And
Yes, I tried to compensate for that.
Yeah.
Yes.
So it was.
go with the question answer session.
Any questions in chat box?
Some question, madam?
Otherwise, I have questions.
Yes.
If there are any questions in chat box, that we will first take. Not yet. They might be.
Hina madam, Hina has posted one question. Hina madam has posted one question in the chat box.
Hina.
Please, madam, please.
No proteins are seen under. It's a question. Just a minute. Yeah. Sir, there is a question from Hina Bojwani. She is our young faculty member and working in the area of molecular modeling.
Great.
Yeah, so she has mentioned that no proteins are seen under three-dimensional visualization, although the sequence has been aligned to some of the proteins from UniProt. But for this cytochrome, it didn't give any 3D visualization based on PDB or DSSP. Any specific reason for this? This is one question.
Yes.
He has also added one more.
Yes.
Yeah, so shall I read everything or you will comment on first part?
First part, let us talk about. So she has said that the 3D structure is not available. So in order to find it out, whether sometime what happens is NCBI is usually shall I say in connection with the 3D database, that is PDB, but many a times the link is not working. So what I suggest you is to copy the primary structure and go to PDB database, and then otherwise in the PDB search box also, because she knows about these things, I guess. So you go to the PDB database and search for cytochrome oxidase, this particular bacteria Pseudomonas. So if you do not get it, it means that nobody is interested in this particular strain, but there is no harm. Pseudomonas cytochrome oxidase is definitely there. If not this particular strain, the Pseudomonas oxidase, I took this strain because the M was there, methionine was there.
Okay.
So I tried to tell you that the N-terminal residue is intact, and therefore you can guess. Otherwise, if it is some other protein, if M was not there, this particular point I could not have mentioned. So I found that in this particular protein, the methionine is there, and therefore I took it. Okay, so what you can do is in PDB there will be plenty of cytochrome oxidase of Pseudomonas, that is for sure. So you can take any of this, and if you want to do a blast, if you blast it, so again you will end up with so many sequences where 3D structure is there. That is, I do not think that you will not get cytochrome oxidase, because it's a very common. You will definitely get it.
Yeah, so
you said that
same thing, sir.
This should also mean that the wild type cytochrome oxidase does. No. Cytochrome oxidase wild type is definitely there. It cannot be. It cannot be because it's a large protein. Everybody has studied it so much detail. The mutants are not there. You can generate mutants, but the wild type is definitely there. Do not. I'm 10 times sure. You just search in this database. Homology modeling you can definitely do, but wild type cytochrome oxidase is there. There is no doubt in that.
So I have one question related to the same thing. When you said that you have chosen, yeah, the wild type. So it was started with M. So that, do you mean to say that it is unprocessed or it is not further, because M is the methionine is the first translated protein amino acid in the protein. So that is why that M is there.
Madam, what happens is, no, it's completely processed only.
Mm-hmm.
But it may happen that I wanted this because there were so many sequences, and in this particular what you call as screen, since the M was there, I wanted to tell you, and I wanted to tell the participants that you should always choose a sequence where it is complete. So how can I tell you that what is complete and what is incomplete? So for that reason, I choose this particular sequence where M is there. If M is there, you can be rest assured that your N terminal is secured. Okay, your C terminal you may not know, but your N terminal is secured. This is how you should filter it, because the NCBI is having N number of sequences. For cytochrome oxidase, if you type, you will end up with N number of sequences. Then how can you be sure which sequence you should take? So this is one of the criteria that you should look into, that your sequence should be complete. It should not be a partial CDS. Sometimes cDNA sequences are also there, but many a times these are partial. If it is partial, so what will happen? You will not get the complete picture. So this is this was done in a short period of time only. I did not do much of what you call quality control, but I thought that I should take this because the M is there. And as you have rightly said, M is the methionine initiation codon, right? For that reason only I have chosen it. Yes, right. You are.
Okay, I think she has mentioned, asked one more question. Any limit on number of amino acid residues? This is regarding the mutant preparation.
No. Great. Very good. Nice question, madam. Madam, I'd like to answer it in this way. So what will happen is, it can happen that because it is very easy to, in terms of the sequence part, that is, these Harvard people have given us so much good software that you can do n number of times of changes. So that you can do, but you should also look for the other quality measures. For example, Ramachandran, if you end up with a three-dimensional structure and your Ramachandran is not good, so there is no point doing three different mutation altogether. So it is always advised that you take one step. Also, there are some more issues. So since given an opportunity, I should also say, so for those who are accustomed with the protein structure and all, so if there is a cysteine residue, please understand, madam, if there is a cysteine residue and in a whole protein structure there are two cysteines. Yeah. So what does it mean? It means that there is a high probability that there is a disulfide bridge. So if you substitute the cysteine, there are possibility that you are breaking the protein to complete destroy, you are making a mess out of it. So do not also substitute cysteines. So like this, there are several thumb rules. Everything is not there in a single book. So you have to keep on reading and understanding things bit by bit so that you acquire more knowledge. So this is one more quality control measure that you should do. You should not, it's a thumb rule, you should not substitute cysteines.
Yes, because it is very important in folding. It must be the folded structure. So you are changing that folded.
Meena madam, there is Vaibhavi Garge to whom the mic is offered. She wants to ask the question.
Okay.
Yeah, Garge madam, you can ask the question.
Hello. Am I audible?
Yeah, you are audible.
Hello, good afternoon, sir. First of all, my introduction is I work as an assistant professor in pharmacology at Bharati Vidyapeeth College of Pharmacy, Navi Mumbai. And frankly speaking, the topic was very new to me, but I could get a lot of information. So thank you very much for the wonderful session. My question is related to the software which we downloaded yesterday, that is Rasween. So are there any specific configuration requirements? Because when I tried opening during your session, somehow I just could only access the main menu. I could not go further into that software. Hello.
Hello, sir.
Sir?
I think he's gone out of the meeting. Wait, I'll just check. No, he's there. He's there. Sir, here.
Sir?
That is what? Yes, yes.
So, should
So
I repeat
this
the
is one
question?
more that. Please repeat, madam.
Can I
Yeah.
request
Please
you to
repeat.
keep your video on, sir, so that
Yeah,
speakers
sure.
can, so that attendees can see you also, and we also need to take one photo with the speaker.
Okay.
Yeah, thank you, sir.
Yes, sir. So my question was related to the software which we downloaded yesterday, that is Rasvin. And when during
yes.
your session,
Please.
I tried to open the Rasvin. I could open the software, but it was not really possible for me to get access through all the menus. I could just only access the main menu. So my question is, are there any specific system related or configuration requirements for this software?
Great question. Very nice question, and it's relevant for many. This is the beauty of this particular software, that there is no absolute requirement. So you can download it even in your Windows mobile.
Okay.
So what happens is, it's a very small package. I think if you give a little more time, you will able to do it all by yourself.
Okay.
It's a very simple tool, and there is no absolute requirement.
Okay.
It's a very simple tool, and if you are having 1 GB of RAM, 1 GB of hard disk, it will run like anything.
Okay. So maybe because of the network quality, because actually in Mumbai today morning it was very, heavy monsoon type of weather. So maybe because of that I could not open it completely.
Yes. There is
It is not
Madam,
web-based.
you please once again reinstall. Yes.
Okay.
Once again re-download and reinstall. Yes.
Okay.
Yes.
Okay, sir. Thank you so much.
Yeah. Sir, I have one question.
Please, madam, please.
Yeah. Sir, now in this whatever software you have mentioned, that is Polyfin 2, it has got a WHESS database. So in this database,
Right.
like, yeah. So first of all, how big database it is, and second thing, can we predict mutations for COVID in this case, the COVID targets with the help of this database?
There are two parts of it again. Madam, you will be amazed to know that there are not less than 500 peer-reviewed papers who have used this particular database to predict mutations for COVID. So people have already done it. There is no point of doing it all over again. Everybody has done it, published their reports, and got their benefits out of it. So the people have randomly used this Polyfin 2 for COVID related research. If you search, you will get plenty of data, so much so that you cannot accumulate everything all together. So everybody has used it, each and everyone who have some interest in COVID. So they did it already. As I have said, that it takes a very small period of time, and you can play with the sequence with it. So everybody used it, and it is most highly used tool for prediction of mutations. And the database, madam, as I am telling you, it's a concise database, and it's dynamic also. Every day they are putting new mutations in the DBSS, the WHS database, and therefore as I have said, the protocol is also adaptive protocol. The software is also changing its structure every day. For example, what happens is 15 new mutation data incorporated. So the style of the calculation will change significantly.
Yes.
So, and it is so adaptive that it is receiving all kinds of input. The writer, if you go to the about page of this particular tool, you will see that, and it is made by genetist as well as computer scientist of Harvard. So Harvard is the university where this Mark Zuckerberg, you name everybody from computer,
Yes.
everybody studied there only.
Okay.
So what happens is they have incorporated each and everything.
Wow.
So it's an excellent database, very, very dependable. But
Yeah.
the thing is, you need to, you need to take the final decision on the basis of your prior knowledge, because they will only give you a probability, not 0 and 1.
Right.
So this is how they have made this database.
Yeah. So there is one more question from Dr. Vrish Dwaj. He is asking, can G6PD deficiency anemia can be treated using alteration in mutated protein?
Great. Excellent, sir, that you have asked about glucose 6-phosphate dehydrogenase deficiency. A very, very important case for clinicians in my area as well as many places in India, where the G6PD deficiency again needs a specific mention of many such things. So what happens is, glucose 6-phosphate dehydrogenase deficiency is an inborn error of metabolism which cannot be treated, and what we can do is some kind of supportive therapy. But here the problem is, the symptoms many a times will be hidden, and it can only be detected if the patient is suffering from some kind of hepatic damage. The bilirubin will be high, the spleen may be bigger a little bit, and if you do a what you call osmotic fragility test of the RBCs, you can find out that there are possibly a G6PD deficiency. So this particular disease itself is a very interesting scenario where mutation has played a much bigger role. But what we can do is, we cannot able to correct it. We can understand the effect of the mutation by the tools that I have described today. But in order to correct the mutation, we should think for gene editing technologies, which will be coming in next 20 years. In next 20 years, we can expect that gene editing technologies by means of CRISPR, a whole new dimension of genomic medicine, CRISPR, which itself require faculties like Madam to propose for a FDP on CRISPR itself, where we'll have where we can have some discussion with it. So other than CRISPR, there is only supportive therapy. For example, the patient is suffering from splenomegaly. The spleen will become larger and he will get rid of the spleen and he will take on antibiotic therapy lifelong. So these are the supportive therapy that we can give. And one more thing, today also I have spoken about paracetamol. So you will be amazed to know that G6PD deficiency cases many a times go fatal due to administration of penicillin as well as this thing, what we call as paracetamol, because paracetamol has a tendency to immobilize on RBCs and there is a drug induced hemolytic anemia. I think sir who have asked the question has all the information to him, and it's a very serious concern and we have done nothing in this regard, and therefore I as a what we call is a person also have come across cases where this kind of situations were there. So what exactly we can do is we can predict the outcome of the particular, but we cannot able to correct it. We have to wait for at least 20 more years so that the CRISPR technology is viable for a therapeutic and a therapeutic reality. It becomes therapeutic reality. This is how I'd like to answer it, sir.
Yeah. Any more questions? Yeah, so I think there are no more questions, but definitely it has given, it has actually, enlightened our thoughts in this area. And I'm sure people will think from this direction, because this is the new area where we need to venture for tailor-made type of therapeutics, and that is where we are now approaching. So, sir, it was a really great session. And thank you so much for sharing all those wisdom of thoughts. So thank you very much, sir. So
thank you, madam. Thanks a lot. Thanks a lot.
Thank you, Dr. Devnath, for being with us and sharing such a valuable information. Thank you.
Thank you, madam. Thank you.
Over to you, Ruchira, madam.
Good evening all. On behalf of entire principal KM Kundanani College of Pharmacy, I take this opportunity to thanks to all those who have directly or indirectly contributed to this today's session. At the outset, I thanks all the eminent speakers, Dr. N.K. Subedar, Dr. Deepak Thakur and Dr. Surajit Debnath for giving such an enlightened sessions. I'm thankful to all the chairpersons, Dr. Mrinal Sanai, Dr. Purnima Hamrapurkar and Dr. Meena Kanyalkar Madam for their motivation and chairing the sessions. A special thanks to all the attendees who have attended the sessions very patiently and have made an active participation in each the session. With these kind words, we move towards the end of today's session. We will meet tomorrow at sharp 9 a.m. for the 4th day of Faculty Development Program. Thank you everyone. Thank you so much.
Ma'am, I'm sorry to interrupt, but there is one announcement which I have to make is that there is a slight change in the schedule. Dr. Smita Shirsagar's lecture was arranged at 9 to 11, which is rescheduled, and instead of that, we will be having a session by Dr. Balchandra Bhange sir from Institute of Chemical Technology. So tomorrow's opening session will be by Dr. Bhange. So wish you, I hope tomorrow also with the same enthusiasm attendees will attend the session. Thank you Ruchira madam for hosting this session so nicely and in a composed manner. Thank you all.